Hello, my name is Candace and I'll be talking about DNA replication. And here's my little diagram. Enzymes called DNA helicase, they use energy from ATP to break hydrogen bonds that hold these DNA strands together. It causes that DNA to kind of unzip. The site where DNA helicase unzips the DNA is called the replication fork. As replication continues, these helicase enzymes, they continue to break the hydrogen bonds, and the replication fork moves ahead of the newly synthesized DNA. Stabilizing proteins, here and here, called SSBP, bind to the single-stranded DNA, holding that strand apart and protecting them from being degraded. So before helicase, ahead is DNA gyrase. When strands start to break apart due to helicase tension, a head is created and knots up the DNA into supercoils. Gyrase then comes in and cuts that DNA and unknots them. It relieves that supercoiling so that it doesn't occur. DNA primase, which is an enzyme, adds RNA primer right here, always in the 5-3 direction. It adds it to both strands. They just travel in opposite directions because DNA strands are antiparallel. Primase adds RNA nucleotides, such as uracil, instead of thymine. DNA polymerase 3, here and here, adds DNA nucleotides like A, T, C, and G to the leading and lagging strands. The reason primer is used is because DNA polymerase cannot add any sort of nucleic acids in unless there's already a nucleotide present. So that DNA polymerase 3, it will continue adding until a stop codon is presented. So just note that the lagging strand is called, this is our lagging strand, and this is our leading strand. Our lagging strand is called lagging because it is moving away from the replication fork, and the leading strand is made in the direction of the replication fork. It's made in one long fragment, not short fragments like the lagging strand. DNA polymerase 1, I think that is right here, this one, cuts out RNA primer and adds DNA nucleotides in its place. So it's going in and cutting that sugar phosphate backbone of DNA. It's removing the RNA nucleotides and adding in DNA nucleotides, completing the exact copy of parental strand complementary copy. These DNA polymerase also proofread to check for any mistakes. DNA ligase, this bad boy right here, kind of is like our glue. So that gap where DNA polymerase 1 cut out that RNA primer is sealed like a glue with DNA ligase. It's a sugar phosphate bond. So in the end, after all is said and done, you have a complete replication of DNA with each DNA having a parental strand and a daughter strand of DNA, also known as semi-conservative.